Hello, everyone, and welcome to today's webinar, Deciphering Cancer Cell Heterogeneity for Precision Medicine. I'm Christina Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Bio-Rad Laboratories. To learn more about our sponsor, please visit biorad.com. Now, we encourage you to participate today by submitting as many questions as you have at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. And with that, I would like to now welcome our two speakers, Dr. Xiaodi Guo, Senior Global Product Manager, Single Cell Technologies, Bio-Rad Laboratories, and Dr. Fatima Valdes Mora, Team Leader, Cancer Epigenetic Biology and Therapeutics Group, Children's Cancer Institute, Sydney, Australia. For a complete biography on our speakers, please see the Presenter tab. Welcome to you both. Dr. Guo, you may now begin your presentation.
Great. Thanks, Christy. Hello, everyone. Welcome, and thanks for tuning into our webinar today. I'm Xiaodi Guo, Product Manager at Bio-Rad. So today we are very pleased to have our guest speaker, Dr. Fatima Valdes Mora, joining us from the Children's Cancer Institute in Sydney, Australia. So today we'll be hearing Fatima talking about her research in pediatric cancer, specifically the epigenomic profiling of tumors and tumor-infiltrating immune cells. She'll also be sharing her tips and tricks in workflow optimization for challenging samples, such as frozen brains. But before that, I also would like to spend just a few minutes to cover some basics of ATAC-seq technology and give you some high-level overview of Bio-Rad's single-cell ATAC-seq solution. Of course, at the end, we'll have some Q&A to address any questions you may have. So hopefully, what we're presenting today can help you make some decisions in designing your own experiment and choosing tools for your epigenetic research. So, well, what is ATAC? Well, ATAC stands for Assay for Transposed Accessible Chromatin by Sequencing. That's ATAC-seq. So in a nutshell, it is a technology to map open chromatin regions across the entire genome. What does that mean? Well, if you look at the diagram on the right, our chromatin can have different states, the open and the closed. So in the open state, where chromatin is more unfolded, it allows the transcriptional machinery to bind to DNA and embark on the gene expression process. So we may consider that open chromatin is more likely to harbor the genes that are actively expressed. And the enzyme that is critical for the ATAC protocol is called transposase. The commonly used transposase is TN5. During a step called tegmentation, the TN5 combines and cut the open chromatin DNA while inserting sequencing adapters. By doing so, it produces a ATAC library that can be sequenced to identify those open chromatin regions. So what do people use ATAC for? Well, since open chromatin suggests active genes and closed chromatin regions indicate inactive genes, by looking at this chromatin state, one can predict the gene activity to figure out what genes are likely to be turned on and what genes to be turned off. And we can also get information about chromatin landscape by identifying nucleosome positions. The ATAC-seq also can provide insights into gene regulation within this open chromatin region, where which regions are likely to be bound by transcription factor and which regions are likely to be promoters and enhancers. That way we can gain a holistic view of the regulatory network. So moving on to the single cell sequencing, there no doubt has been a growing awareness of cellular heterogeneity within our tissues and within the tumor samples, just as illustrated by this analogy. I think it really speaks for the power, the importance of single-cell sequencing. So imagine our tissue or a tumor is like a bowl of fruits. The bulk genomic is like making smoothie out of them. It averages out all of the populations, yet misses out important information of the differences among individual fruits. And this kind of cellular heterogeneity can only be dissected with single-cell genomics. So why do people do single-cell ATAC? Well, since each cell can have different chromatin state, this so-called differential chromatin accessibility can help researchers identify cell types, cell subsets, and cell states from a mixed population or a complex tissue. When it is applied to the cancer epigenomics, which apparently another rapidly growing field, and Fatima will be talking about in a moment. So with single-cell ATAC, researchers can study what impacts a drug might have on individual cells to their chromatin states, and also identify potential epigenetic biomarkers or drug candidates. With those information, we can stratify patients for more personalized medicine. So Bio-Rad, as a pioneer in droplet technology, we also brought our own single-cell ATAC-seq solution to the market, aiming to provide a cost-effective yet high-performance single-cell ATAC solution to researchers. Starting with our DD-seq single-cell isolator, which is a droplet-based single-cell isolator and enables fully automated and fast single-cell partitioning and single-cell barcoding. And it does it only in just about 5 minutes. Our SureCell ATAC library prep kit, which provides reagents to process up to 8 samples with a scalable throughput from hundreds to thousands of cells per sample as an output. Once you get your sequencing data, you can move your data to our single-cell analysis software, the Omnation, for a streamlined QC, data analysis, and reporting. It also has the batch processing and batch reporting functionality to process multiple samples in parallel, which saves a lot of time. The output file of the Omnation could be plugged into commonly used tertiary analysis software, tertiary analysis tools for further data mining or biologic interpretations. So our assay is just needs a single-day workflow. It's easy single-day workflow from the starting materials to sequencing-ready library. It takes less than 8 hours with 2 safe stop points. And the protocol supports both whole cells and cell nuclei as input, which provides certain level of flexibility when you work with different sample types. So as one of the benefits of the ATAC technology versus other canonical epigenetic approaches, you can really use much fewer cells to start the workflow. Now for our assay, you can start with as few as 150K cells, which already accounts for the cell loss during the washing, the counting, and the nuclear prep. Last but not least, the superior cell utilization, the industry-leading capture efficiency we can deliver between 60 to 80%, sometimes even higher. This is made possible by our droplet technology and our innovative bead merging approach, which allows overloading of beads to maximize the chance of cell bead pairing. So we talked about the simple workflow and high efficiency, but the DD-seq ATAC solution also delivers exceptional data quality, especially for those ATAC metrics that speaks for the assay sensitivity and specificity. This includes the TSSE, the transcription starting site enrichment score, and the FRIP, the fragment in peaks, both of which have been constantly shown comparable or even better than other commercial and lab-developed protocols, as reported by our customers. We also have excellent crosstalk rate less than 5% at 5K total output. So our protocol has been tested internally and externally with a variety of sample types, including human and mouse cell lines, human and mouse primary tissues, and primary cells. So in the interest of time, I'm not going to talk about too many details and data. If you find this is something might be interesting for your research and you care to learn more, please type more info in the chat box. One of our single cell specialists will reach out to you as soon as possible. So I'll stop here and pass it over to our guest speaker, Dr. Fatima Valdes Mora. Fatima, all yours.
Thank you, Shawty. As Shawty mentioned, I'm one of the team leaders at the Children's Cancer Institute, and today I'm going to present an overview of epigenetics, just to an introduction about why performing this type of analysis, as Shawty mentioned, is important, and in particular in the I will also talk about the different single-cell epigenetic methodologies that are available so far. I will focus on talking about the single-cell ATAC-seq method, mainly based on the source cell from solution from Bio-Rad. And then I will present two case of studies that we've been doing to look and to tumor heterogeneity, in particular focusing on the tumor microenvironment using this technique. So just to quickly give you an overview of epigenetics, epigenetics the prefix epi from the Greek word above, so it means above genetics, and it comprises different molecular processes the cells use to actually control gene function without altering the DNA sequence. So here in this slide, you can see summarize the main different molecular mechanisms including DNA methylation, modifications, non-coding RNAs, and so on. But in particular, I wanted to highlight the chromatin accessibility and positioning. So all of these modifications work together to alter the structure and the functional particularities of the local chromatin to end up regulating gene function and therefore cell fate. So if we want to study all of these different epigenetic processes, we have different techniques that integrate all of these, each of these processes at the genome-wide level. So these are epigenomic techniques, and they're summarized here, as you can see, in particular to interrogate chromatin accessibility to see which regions are open and closed, and that translate in gene transcription. ATAC-seq is one of them. So attention epigenetics is very important in the regulation of gene function, and this is very important for its normal development and for the determination of cell types. But what happens is that in cancer, genetics is profoundly altered. And even though we see cancer as a genetic disease, it is also an epigenetic disease. Because when in the process of carcinogenesis, the chromatin undergoes profound changes, and this is an example to show you what kind of changes we can see. So certain regions that were open in a normal cell become closed chromatin regions, and therefore results in gene inactivation, as you can see on the top part of the chromatin outlined here, or other regions that were closed and they have to be closed for normal functioning of the cell become open, and this allows the expression of oncogenes and other aberrant genes that help cells to become cancer cells. So as you can imagine, this is epigenetics is a very important process to understand during cancer development and progression. And I wanted also to highlight here that the epigenome of the cells in cancer is not only altering cancer cells, it is also altered on the cells from the tumor microenvironment. This includes immune cells, stromal cells, and endothelial cells, help the cancer cells to actually grow and progress and metastasize. And these cells don't carry any genetic mutations. They actually alter their functionality by interacting with cancer cells and by altering the epigenome of these cells. So that's why it's very important to actually understand how the epigenome changes in each of the cell types that participate during cancer progression to find which therapy will be the best for each particular cancer. So how can we actually tease apart the different epigenome of all of the different cell types in tumors? So as you can imagine, the best solution for this is to perform single cell technologies. As Charity mentioned with the smoothie effect, with single cell technologies, we can tease apart the epigenome transcriptome, in case we do single cell RNA-seq, of each of the different cell types that compose the heterogeneous tumors. And one of the things that I wanted to highlight here is that single cell RNA-seq has been the leading technology to tease apart cell types and cell functionality in tumors so far. But as I mentioned, because the epigenome is altered not only in cancer cells, but also in the cells from the tumor microenvironment, it's also very important to investigate how the epigenome and the transcriptome changes in these cells. So with that, I wanted to start talking about what different methods have been created to interrogate the epigenome at the single cell level. And with this, I just wanted to highlight the different methods according to the date of publications and also reflect the high throughput of these methods. And as you can see here, in the early 2014, techniques like single cell Hi-C and single cell bisulfite sequencing were introduced, but the high throughput amount of cells that were interrogated was quite low, and it was highly costly. So if we go to 2019 and 2020, we can see that another technologies to interrogate the epigenome were introduced and at a higher capacity of number of cells. And as you can see at the top, one of the top ones that is highlighted here is the single cell ATAC-seq. And this is a method that interrogates the chromatin accessibility and the different chromatin stages of the cell. And as you can see, we can reach about 1 million cells per in terms of numbers of cells that you can interrogate from a sample. So this is very important when we want to explore the heterogeneity of tumors. Having a representation of 100 cells from a sample is not enough to get the actual resolution of this heterogeneity. So we need high throughput methods to interrogate this. So with this, I wanted to focus on the single cell ATAC-seq solutions that have come up with that offers the option of high number of cells, and they're quite cost effective and quite quick to do. And as I mentioned, the SURCell ATAC-seq kit is one of these solutions and is based on this Nature publication in 2019. And this here, I have summarized the molecular workflow that follows this procedure. It's a based protocol where you have bits that contain a PCR handle, which is common to all the bits, a cell barcode, which is different in each bit, a unique molecular identifier, which is unique for each of the primers that are attached to the bits, and then a TN5 complementary sequence. So through this droplet generator that you use, in this case, use the DD-seq droplet encapsulator, you put the bits through one of the, and from the other one you put your cells that have been dissociated and the nuclei have been isolated and transposed. And once the droplets are generated, you will have here, as you can see in the droplet that has been generated, the bits containing a nucleotide that has been transposed. So by doing a linear PCR in the droplets, you are able to copy the accessible DNA sequence and the TN5 sequences flanking these accessible regions into the bits. And what happens in the droplets, you break the droplets and you proceed to do PCR to generate your library of the ATAC-seq samples. So once you've done this, which Sarati mentioned, it takes about a day to perform this, then you to do library sequencing. And as I'm going to talk about this later, so far this ATAC-seq was offering using the NextSeq 500 sequencer, but as I will explain today, we scale up this to other platforms that are more high throughput. And once you get the data, you will be able to generate your pickle digital count matrix, and you will be able to perform your clustering and visualization of your data. Of the advantages of ATAC-seq, of single-cell ATAC-seq offers, is that you will be able to do more analysis than the transcripting, because it will not only include the determination of the different cell types based on the peaks that have been called in its cell, but also you will be able to have the different chromatin stages you obtain after performing this. So the workflow of the bioinformatic analysis can be split in two. First is the QC and the metrics, just to see that your experiment went well. And the second are the tertiary analysis that you do to actually interpret your data and to understand the data that you have in front of you. So for key analysis, we have one of the analysis software that Omniscient, that has been created by Bio-Rad. This software will give you all of the standard metrics just to see that your experiment went well, the TSS enrichment score, your fragment profile of the fragments, the normalization and batch effect correction, which is very important, as we observed previously that there's a lot of difference with bats. There's a lot of batch effects in this process, and then it will generate. And then the next step will be generating the peak coding of feature digital matrix, and then after this, you will get the classical UMAP with the dimensional reduction cell clustering. So the tertiary, the kind of downstream analysis that we can perform with using different bioinformatic tools, including Omniscient, is to link this regulatory network that we have observed with ATAC-seq, with the open regions not farther than transcription start sites. You can link to genes and you can call different cell types. You can perform motif analysis just to look at transcription factor binding sites that are possible. You can perform interaction prediction between promoters and enhancers just to link this, create this gene regulatory network, just expression of a gene, but also the presence of enhancers. You can also perform pseudo-timing analysis to see if this kind of generation of cells that are kind of becoming different cell types, so they come once from the cell. And you can also integrate your single-cell ATAC-seq data with single-cell RNA-seq to integrate the resolution of your cell type, but also to link the expression of transcription factors to the presence of open regions, and link this to create this big network for each of the cells. So there is not as many bioinformatic pipelines for cell ATAC-seq compared to single-cell RNA-seq, but this is becoming more and more less than a problem, because there's more bioinformatic tools being created in the last few years. And this is just a summary showing what's like a comparison of different methods that have been published to do the QC metrics, but also this downstream analysis. And just the ones that we have been using quite nicely and easily we found very useful are scATAC and ARCH-R. And I put here the reference for if you want to check how these pipelines work. So once I've done the introduction of single-cell ATAC-seq and why it's important to use this technology to interrogate the cancer epigenome, I just want to talk about two cases of study that we've been working on in the last few years in the context of the tumor microenvironment. So the first one is analyzing rare tumor-infiltrated Milo immune cells that we believe that contribute to tumor progression. So we wanted to see if we could use ATAC-seq to determine how these cells were changing according to malignancy, cancer, and also the progression of the cancer through metastases. And because of the fact that these cells are rare and are infiltrating tumors but are rare, we wanted to see if we could use ATAC-seq in fact sorted cells and not just from the unbiased tumor sample. So for that, we used a mouse model of breast cancer. There's 41 and 67 in our mouse models, which are two models that represents triple negative breast cancer. So the difference between these two models is that the 41 is a metastatic model that metastasized to the lungs, and the 67 are non-metastatic model. We use these two samples, so we basically isolate the cells from the lungs, which is the metastatic site on the malignant model, just to see if these cells look different to the scenario and if these cells somehow have phenotypic features and epigenomic features that show that they were contributing to metastasis. We also use 2 time points, 2 weeks, which is at 2 weeks, these mice don't present any metastases in the case of 41, and is development of metastases. So basically, we wanted to see if these cells were there already in this where the metastases were not there, that they were somehow contributing to the establishment of the pre-metastatic niche. So we basically the protocol, this protocol that we published in 2021 to digest the tissues and fact sort these Milo cells. And here I have given you a summary of how we sort these cells. For this is unpublished data, so I have used A and B as additional markers to isolate these cells. With this fact sorting, we enrich these Milo cells containing in the lungs. And then with this, we perform single cell using the Bio-Rad solution system and the single cell RNA-seq. So this is like 2 different samples done in different timings. So the idea was to obtain more information about how these are rated in terms of chromatin stages and how this can be correlated with the transcriptomic states of the cells. So just to give you an overview of the quality of the single cell ATAC-seq as a general QC, we observed the classical tape station profile of a transpose samples. In all of the samples, it looks really nice. And in case of the QC measure, it looks pretty nice. We aim to capture around 5,000 cells, and we obtain a good number of unique nuclear fragments per cell, line reads, expected number or a percentage of duplicate reads, and expect enrichment of DSSs of the reads at the sites. And with that, we proceed to do the downstream analysis just to determine the phenotypic features and epigenomic features of these cells. So just to give you a summary of what we found is that first, this is a UMAP doing like unbiased kind of UMAP. We see that these cells in general, they don't look too different to each other. Expected because they are a subgroup of Milo cells, but we do see different clusters coming up in the, as you can see, 1, 2, 3, 4, 5 different clusters. If we look at the transcriptomic and epigenetic features and the literature of the functionality of these cells, we found that these from all of these cells, we found that the green ones correspond to non-malignant Milo cells, so the typical immune cells that are there to protect the body. But then we found a subgroup of types of malignant cells, malignant myeloid cells that are the red ones, and metastatic myeloid cells that are the ones in the brown. So we how did we find this? Basically, what we found and how we find this is that when we could look at the metastatic breast cancer model, the 41, we could see that these metastatic myeloid cells are present at 4 weeks. So that's suggesting that they're involved in the metastatic process, and they were not present in the non-metastatic breast cancer model, the 67NR. While these cells, the malignant myeloid cells that probably are responsible for immunosuppression in the primary tumors, were present in all of the samples except for the metastatic one, which might be involved in the cancer progression, in the cancer growth, but not in the cancer metastasis. We also use our RNA-seq data just to see correlate the ATAC-seq data with the RNA-seq data, as you can see on the top right. And this is just an example that is highly expressed in the metastatic myeloid cells. And we can see that from the point of view, the peaks are more accessible, the chromatin is more accessible at the promoter of these cells, but also we found an cancer close by that is highly accessible only in the metastatic myeloid cells. So as a conclusion of this study, there's a few conclusions, technical and also biological. The SURCell ATAC-seq kit can be used. We can use this method in FACS-sorted tumor-infiltrated myeloid cells that are not highly abundant at tumor prep. Using this ATAC-seq, we are able to see heterogeneity within myeloid cells according to tumor state, as I showed you in the HUMAP. And also, I haven't showed you this, but we have a very, very high correlation between the single-cell RNA-seq data and the single-cell ATAC-seq data. And also the integration that between RNA-seq and ATAC-seq data allow us to build a more comprehensive gene regulatory network to unveil key processes for suppression with these malignant cells, and also for cancer progression and metastasis with the metastatic myeloid cells. So the next case of study that I wanted to talk to you today is a study that we've recently performed using flash brain tissues. So we aim to perform single-cell ATAC-seq from these tissues because from our point of view, the protocol was, we were not sure if we could apply this protocol into flash frozen tissues. As a standard, you need a high viability of cells to start with this protocol, and we were not sure if there's a possibility when you start with flash frozen tissues. So this is an experimental workflow summary of the samples that were collected. So we have a pediatric brain tumor where from the same patient we collect from the peripheral context, we collect normal brain, and then we collect the primary brain tumor, which was localized in the pons. We also collect an local metastasis in the cerebellum. So we have these 3 samples that were flash frozen, and they were stored at -80 for certain few months. So our idea of this was to check if we could use SURCell-seq to perform single-cell ATAC-seq from these tissues. That seems to be obviously more challenging than the data that I presented previously, which comes from fresh tissue and FACS sorted. And when you do FACS sorting, you actually select viable cells. So I guess the main challenges for us in this process was to know if the critical step was the nuclei prep. So we have to make sure we get rid of all cell debris and of cell death that could contaminate our single-cell isolation process, single-cell nuclei isolation process. And also the other part that we wanted to explore using this method at the same time was to see if we could use another sequencing platforms that allow us more cost-effective and high throughput more than NextSeq 500. So for us, the most important point to make sure we succeed in the single ATAC-seq protocol was to have a good and highly viable nuclear. And to do that, we couldn't find a specific protocol that will help us, especially coming from brain tissues, and we combined protocols just to see if we could, using the different suggestions from each protocol, we could build up our own protocol based on our previous experience with this kit, the single-cell ATAC-seq kit. And this is just to give you an example of the main QC that we perform, is just to visualize that we could see a clean single-cell nuclei prep. And this is just an example of one of the samples that we perform at the nuclear preparation. So basically, we took the tissue and do single-cell dissociation, and then perform a gradient to get rid of all of the dead cells, and then we perform like a nuclei prep, and again a gradient for samples. And this is how the sample looks like prior to cleanup. And as you can see, it's really messy, and it's full of cell debris, of cells everywhere. After our nuclei preparation, what we got is a very nice nuclei that's stained with trypan blue, see that they were permeable and they were accessible for transposition. And as you can see, the sample is very clean. So we were happy. We performed the transposition, the single cell, the single nuclei droplet encapsulation, and the transposition, the nuclei. We could see a nice tape station profile as expected for a single-cell ATAC-seq protocol, and so we were quite happy with the library prep. So the next steps that we did is just prepare like a protocol to prepare the samples for NovaSeq 6000. This is the summary of this particular experiment, where we index 3 samples at 5,000 cells. We get between 20 to 30,000 unique reads per cell, and that which means that we're using NovaSeq SP200 cycles format at the sequencing here 118 read 1, 8 for index, and 40 for the read 2, and using a custom primer one. We didn't use a 5x spike control because the complexity was good enough from our library samples, and this is the that we loaded our library based on our previous experience using bulk ATAC-seq samples in NovaSeq 6000 and 260 picomole. So with that, we perform the sequencing. The standard QC from the sequencers look really good. We ran the Omnition software just for the QC metrics. We could see that everything looked pretty good in terms of cell number that we aim about 5,000. So we got around 5,000, 7,000 for one of them, and 3,000, which looked pretty good. The median fragment, unique nuclear fragments looks reasonably good, and also duplication rate and percentage of reads in the transcription star site look as expected. So we also created our UMAPs using of each of these different samples independently using the Omnition software. And we could see that as expected for a tumor sample that contains not only cancer cells but stroma cells and immune cells, we could see that there were different clusters that separate quite good between in each sample and between samples. But the next step to do is obviously compare the difference between the normal brain from this patient to region from brain, primary tumor, and the metastasis. So we have to actually integrate this data together. So for that, we use ArchR, and this is the integration of normalization and integration of the data. And as you can see here, the red is the local metastasis, the blue is the normal brain, and the green is the primary tumor. And this looks like the sum clusters that are specific for each sample, but other clusters that are actually come from all of the samples together. We identify where each of the clusters were in terms of cell types. We found that the clusters that were combined with samples respond to immune cells as expected. These are Milo cells in the cluster number 3 and T cells in the cluster number 5. And the cluster number 2, that seems to be very specific for the tumors and the local metastasis, corresponds to malignant cells as expected. And the cluster number 4 and the cluster number 1 correspond to normal epithelial, normal astrocytes and oligodendrocytes that are coming from the normal sample, which is expected, and that was very, very nice to see. Just we're still working on this data, make sense of this data, to highlight one of the main discoveries that we've made with this sample using ATAC-seq, which is that when we look at portions and the presence of each of the cell types that we identify, we found that T cells were quite deleted in the tumors. As you can see, all the T cells that were detected were present in the normal cell, not the tumor or the metastases, while the Milo cells that were identified are mainly, especially in the local metastases, and followed by the tumor, and they were less predominant in the normal tumor, in the normal brain. So as a summary of the discoveries of the case study 2 is that we can say that SurCell ATAC-seq kit can be used in frozen brain tissue samples after sample cleanup and nuclei isolation using a protocol that we've developed combining 3 different protocols that have been published. That samples from the SurCell ATAC-seq kit can be sequenced using another sequencing platforms, 6000. And this ATAC-seq data reveals that brain tumors and brain tumor heterogeneity can be identified, and we can identify differentially represented cell types depending on the tumor, on a normal, or on the metastatic status of the sample. And with that, I would like to finish by acknowledging everyone that has been involved in these studies. It's been a few years working on single-cell ATAC-seq. The people of the group, our collaborators that have enabled the sample, obtaining the sample and also processing the samples, and the Bio-Rad R&D team that have been always helping us with all of the troubleshooting that we had with the and also with the sample analysis, experiment analysis. So thank you very much for listening, and I will be happy to take any questions.
Thank you both for your informative presentations. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located at the bottom of your screen, and we will answer as many of your questions as we have time for. Additionally, if you would like to learn more about Bio-Rad's single-cell ATAC-seq solution, please type more info into that question box, and a specialist will reach out to you soon. Okay, let's get started. We have lots of questions coming in. Let's see. Let's start with this question. Dr. Valdes Mora, let's start with you. Can you explain how the ATAC data might be impacted by suboptimal nuclei prep?
So as I mentioned in my talk, it can be highly impact if you don't have a proper nuclei prep. There is several points that you have to consider. The first is that the cell debris from dead cells, if you don't remove those, that will definitely contaminate your sample, and you will get a lot of sequencing data that doesn't come from a cell, is coming from floating chromatin that comes from dead cells, and that is going to really impact on the quality of your data. And also, if it's very important to have nice permeable nuclei prep to make sure that the transposase is able to access the chromatin and perform a good cutting of the accessible sites. So I will say that is the most important part of the protocol, to make sure that you get that right. If you get that right, everything suits smoothly.
Thank you. And Dr. Guo, let's go over to you for our next question. Is the Omniscient software different from the Bio-Rad ATAC toolkit?
Yes, absolutely. So the Omniscient is our single cell, we call it a single cell data analysis hub. It was launched last year to replace the Bio-Rad single cell, or Bio-Rad ATAC toolkit, but we're also going to keep building it as we're rolling out more assays. So compared to the toolkit, the Omniscient really had a lot of improvements in utilization, in workflow efficiency, and functionality. For example, as I already briefly mentioned in my talk, it can process multiple samples in parallel and report multiple samples, and it can also take combinatorial indexing data, especially the DSCI ATAC that was published by also Jason Birostro's lab in 2019 Nature Biotech paper. So yeah, a lot of great improvements compared to the original toolkit. We definitely encourage a customer to start using it as soon as possible.
Thank you.
If you have it.
Thank you very much. Now, Dr. Valdes Mora, the data is convincing for a myeloid cell in the tumor. Have you looked at the lymph nodes and observed the same pattern?
Yeah, we've looked at not lymph nodes, we've looked at spleen and blood, and we do see that those myeloid cells, when there's a tumor and there is a metastasis, are still there. But this suggests that there is kind of some trafficking of the myeloid cells from these organs to the tumor and all vice versa. And it's something that we are further exploring using single cell methods.
Thank you. Dr. Guo, we'll go over back to you. This question has come in a few times. How many cells are required for the Bio-Rad ATAC assay?
Right. Yeah, we and also mentioned briefly in the talk, we need as few as 150K cells to start the workflow. And that already accounts for the cell loss during the nuclear prep, during the subsequent washing and counting steps. And then once you isolated the nuclei, you need at least 50K or 60K nuclei for the tag-mutation step. Having said that, we have tried fewer nuclei to continue with the workflow, and fewer nuclei and fewer cells has been demonstrated. I think you can go lower or go higher. Really, it's about how you modify the protocol to suit your sample type as best as possible, as good as possible. So, yeah, thanks for asking.
Sure, very good. And we'll stay with you for this next question as well. Can you explain what samples could use whole cell as input, and what should the nuclei?
Sure.
And what should use the nuclei?
Sure, yes. So we have showed, well, we have two protocols, basically, we're supporting two protocols. One is called Almini protocol. It's using the isolated nuclei as input. So basically, you use slightly harsher detergent to isolate the nuclei, and then proceed with the downstream steps. And then that's good for cell lines, or we have tested this protocol with a lot of cell lines like 293, K562, NH3T3, and other GM12878 cell line, etc. And then for some more fragile cells like the PBMCs, like the bone marrow mononuclear cells, we will recommend using the whole cell protocol, which uses slightly gentler the lysis buffer, I would say, so to process them. It will work just beautifully.
Very good, thank you. Now, Dr. Valdes Mora, over to you. We have an attendee saying, great work. And do you think FACS sorting could possibly have an impact on cell biology? And if yes, how to minimize that?
Yeah, that's a question that keeps coming up for single cell methods. Yeah, I agree that FACS sorting, I mean, the whole tissue dissociation and single cell isolation might impact in some of the biology, but the essential of the cell types specification is not really altered. Like you still see the different cells that are present on these cells on these samples. FACS sorting it could affect the prep, but we've actually have done the same experiment not doing FACS. So we basically just took the whole tumor and performed single cell RNA-seq. We see a very, very nice correlation on, if we focus on these FACS sorted Milo cells, they look very, so they don't seem to be affected by the FACS sorting, which is a really nice thing. So yeah, I think based on our experience, FACS sorting is a really nice way to isolate cells and to get rid of dead cells. That gives you a really nice prep to perform single cell experiments. And if the cells are not too fragile, so maybe I will be a bit worried with neurons, but if the cells are not super fragile, they are fine with FACS sorting. So it's fine.
And Dr. Valdes Mora, let's stick with you on this next question. Do you recommend using DNase 1 treatment for low viability samples?
Yeah, absolutely. I would say that when the viability, you check the viability of your cells and it's below 85% or even like maybe 90%, I would definitely do DNase 1 treatment. But if your viability goes below, let's say, 75%, DNase 1 is not enough. So you have to do other procedures like gradient centrifugation, just to isolate the viable cells from your prep. But definitely, if your viability is between 90 or 85 to 75%, the DNase 1 is a quick and easy step to add to clean up your sample.
Very good. Thank you. And Dr. Guo, we'll go over back to you. Will the output files from the software be compatible with Archer?
Oh yeah, absolutely. Well, Fatima already showed her beautiful data analyzing Archer and scATAC, right? So yeah, the output file from Omnation can definitely be plugged into those commonly used tertiary tools. Bio-Rad, we actually do not really support those tertiary tools, but we do have dedicated Bioinformatics FAS who are always eager to help our customers in their data analysis. So if you really run into any troubles or you want some guidance, yeah, feel free to email our tech support, and we're happy to help you as much as we can.
Very good, thank you. And now we do have time for just maybe 2 more questions. Let's stick with you for this one. Can the Bio-Rad ATAC kit use fixed or preserved cells?
Preserved, well, if it's cryopreserved cells, absolutely, right? They cryopreserved cells, the brain's tissues, for example, we have a lot of customers using those samples. But for fixed sample, we haven't exhaustively tested samples that are fixed yet on our protocol yet, but our customers have recommended lightly fixed cells with such as 0.2% paraformaldehyde, and it worked well with our assay. But I would say you should expect some little bit higher background or lower library complexity, and maybe lower intensity of peaks that might be expected with fixed samples. So just as with any, dealing with any new sample types, you need some kind of optimization to make sure it works the best.
Very good. Thank you so much. And let's wrap with this final question. Dr. Valdes Mora, what will be logical next steps for your findings? So other epigenetic technologies or knockout models?
For us, the next step is continue with the analysis of the data, because what I've shown today is actually the first steps, but we want to further utilize the single-cell ATAC-seq data that we have to further explore more things. Like, as I mentioned in the summary of the tertiary analysis that we can do, is explore transcription factor usage, combine this data with single-cell RNA-seq to increase the resolution. So we will continue mining the data and continue doing more analysis, because this analysis that I showed today just is the tip of the iceberg. We can do so many things with the data that we already have. So next natural step will be expanding the analysis to get more out of it. And the next step will be combining this single-cell ATAC-seq data with other single-cell technologies to obtain this comprehensive single-cell multi-omic data. And also, one of the things that are quite interested, especially with the infiltrated immune cells in tumors, is to perform some validation in terms of the location of these cells within tumors. So either performing classical immunohistochemistry assays to see the localization, or go a bit, like, depending on the budget, but perform spatial transcriptomics on the same samples.
Thank you both so much for your time today and for your important research. I do want to thank Collaborators, but also our sponsor, Bio-Rad Laboratories, for underwriting today's educational webcast. And before we go, I do want to thank our audience for joining us today and for their interesting questions that we did not have time for today, and those submitted during the on-demand period. They will be addressed by our speakers via the contact information you provided at the time of registration. I know we mentioned this before, but again, if you do want to learn more about Bio-Rad's single-cell ATAC-seq solution, please type more info into that Q&A box, and our specialist will reach out to you soon. This webcast can be viewed on demand, and Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thanks for joining us. Until next time, take care, everyone. Bye-bye.
Bye-bye. Thank you.